Welcome to our second unit of this module 3 breeding methods fundamentals. This unit 2 is aimed at common methods and strategies in organic breeding. Pedro, Barbara, and me will be the co-presenters. For those who have not come at the first unit, I repeat, this module, this training activity is one of the different training opportunities offered by LiveSeeding project. For more info, you can go to the training option of the LiveSeeding webpage. We are specifically within the organic plant breeding training, the one marked in red, but there are many other opportunities in treating different issues related to organic breeding and organic seeds, organic cultivar testing, high quality organic seed production, regulatory and policy aspects of organic seeds, entrepreneurship in the organic seeds and breeding sector, embedding organic seed and cultivated diversity in city food policies, and finally the summer school. So you have a range of opportunities in training within the LiveSeeding project. But now we are with the organic plant breeding option. As I told in the first unit, today we are treating with module 3, breeding methods fundamentals. We already launched module 1, plant genetic resources, and module 2, phenomics, in the past weeks. And the upcoming module 4 and module 5 at the beginning of March 4th and March 7th, module 4, development and application of molecular methods in organic breeding and organic heterogeneous materials, design and development, will be the last ones at the beginning of March. But today we are on module 3, breeding methods and fundamentals. We have already showed unit 3.1, unit 1 within this module, the generation of new diversity. We come from the break, and now it's time for unit 3.2 or unit 2 within this module 3. Unit 2, which is aimed at common methods and strategies in organic breeding. I repeat, Pedro Mendes, Barbara Pipan, and myself will be in charge of this session, this unit. Then at the end, we will, about 1, we will make a break till 2:30, when we will restart for unit 3, calculation and evaluation of key breeding parameters, and finalizing by unit 4, fundamentals and participatory plant breeding. Well, as always, we start one of the units. We prepare a short conceptual index for this unit, the second unit of today, breeding methods. We will perform a mixture of presentations about the main topics on common methods and strategies on organic breeding. Basics, the primers, the basic knowledge, and practical examples. Okay, I will present the basics next. Then I will give the floor and the screen sharing to Pedro. And after Pedro will be the time for Barbara. At the end, we will perform, as in unit 1, a fast quiz, which will take about 10-15 minutes. And at the end, we will perform a debate, answering question, and grow up and propose a work homework, which will take about also 10-15 minutes. Okay, I repeat, as on all the units during the presentation, we are delighted in answering your question, but please put them through the chat. And Petra will manage and will talk us about the the questions and she will manage. It's a more efficient way than giving the floor to people asking, etc. Okay, because we are too many people, so it's more efficient putting the question through the chat. Okay, and also important, as always, marked in red, important for certificates in all the units is common. To get the certificate, it is important to fill the quiz, okay, and put the answers in the body of the mail. In the case of this unit, please, the quiz should be answered to Pedro, PMM ESAC PT of Portugal, my email, and very important because she's the first checking to Petra's email, okay? And the same for the homework, which will be proposed for next Thursday. So you have till next Thursday to finish the homework, okay? And please remember to these 3 emails, the one from Pedro, PMM, the mine, and important Petra's email for both the quiz and the homework. Well, let's start from my part, which is the basics and basic knowledge of methods and strategies. A first review. I don't want to bother you with hundreds of methods of breeding. Actually, there are a few quite common, and the rest are adaptation or mixtures of these 4, 6 common methods. Okay, so my part of the presentation will be very fast and very simple, putting emphasis in the most important strategies and the knowledge. Well, first of all, you must know that the reproductive system of the species you are working with is essential for breeding purposes because it defines, it limits the genetic structure of the population you will be working with. At the moment you are starting, because this population comes from land race population variety, traditional land race past, okay, and what I have to start now, but also will define the reproductive system of your species, how this genetic structure evolves during the breeding process. Okay, not only what you have at the beginning, but also how this genetic structure evolves from the beginning to the end of the breeding process. Okay. In this regard, probably people familiar to breeding concepts, but maybe not the others, not the other attendant people. When in the case that your species is an highly autogamous species, the genetic structure equilibrium of this kind of species goes to homozygous populations. I mean, what you can find in a population variety of an autogamous species is usually a mixture of inbred lines, because they come from open pollination. They have been evolved in the traditional breeding selection of the best individuals by the breeder. They are usually self-fed, season by season, year by year, etc. And at the end, what you have should be a mixture of inbred lines. Otherwise, what you have is the beans of Mendel's beans, which were directly highly inbred lines of peas, which were highly homozygous. So your variety can be an unique inbred line, highly homozygous genetically, or maybe a mixture of inbred lines. It depends on the past stages of the history of this population. Okay, but if this is an autogamous species, the equilibrium should be more or less like that at the level of the genetic structure of the population. In the case of allogamous crops, their own nature, reproductive nature, to be open pollinated evolves or makes that this population evolves towards mixtures of homozygous and heterozygous individuals, and also individuals which are a mixture of everything. Some genes are homozygous, and then they are inbred lines for these genes, but while for other genes they are still heterozygous. So at the equilibrium of allogamous crops or species, what you should find usually is a mixture of everything, usually accomplishing the Hardy-Weinberg law. Okay, so a mixture of individuals which are a mixture of genes in homozygosity, genes in heterozygosity, individuals we have most of their genes in homozygous and a small part in heterozygous, or the contrary in others, so a mixture. And they are usually more heterogeneous than autogamous species, okay? Mainly due to this allogamous, open to cross-pollination nature of the reproductive system. Okay, and finally we have the vegetatively reproduced species. These species, or the varieties, or the population varieties of these species, are usually clones, because they are so heterozygous that the only way to ensure that the offspring fits the phenotype of the variety is cloning them, is reproducing the individuals vegetatively from the mother clone. So when you are looking at the orchard of one variety of Valencia late citrus, or one peach trees orchard, you are seeing a population usually, I repeat, usually of the same genotype, which has been cloned. Okay, maybe your farmer has an orchard where he has mixed different clones, different varieties, because I like very much this peach variety, and these two citrus varieties. Okay, they have 4 clones, each one reproduced 10, 20, or 100 times. Okay, but these species are vegetatively reproduced because there is no other way to ensure that the phenotype, the good performance of the variety is preserved, but cloning. Okay. Well, also related to the reproductive systems, it is also important considered in those species reproduced sexually, the first two, autogamous and allogamous species, is the inbreeding depression. Okay, inbreeding depression appears when you are selfing too much the individuals of your population, or your population is submitted to, in some part of its history, is submitted to a genetic bottleneck where the genetic diversity is too narrow, and then many of the individuals are quite related, and then there's no other opportunity that crossing among them. Okay, or during your breeding process, you are forcing selfing, okay, to ensure highly homozygous individuals. This is the problem in breeding depression, which this selfing forced towards highly homozygous lines usually provokes the appearance of deleterious syndrome or deleterious performance of the individuals. Okay, in humans or in animals is quite common. In plants, they are more resilient for that, but problems of deleterious or weakening syndromes may appear. But we must make two differences. In breeding depression is not usually a problem in autogamous species because during their domestication and breeding, the history of these species, domesticated species, they have been submitted to narrow bottlenecks or they have suffered previously these syndromes of inbreeding depression. And now, centuries after or millennia after, those genes which provoke inbreeding depression have barely disappear from their genomes. So in autogamous species, it's not a problem to force selfing, to force inbreeding towards highly homozygous lines, okay? But the problem is quite usually in allogamous. Mice is an example. If you inbred 1 or 2 generations, Pedro will provide more details, a variety of mice, and then compare the original variety with these inbred lines, you will find that these offsprings are weaker, are smaller, do not feed and produce big big grains, etc. So it's quite evident and illustrative. So inbreeding depression is not usually a problem in autogamous, but is usual in allogamous. And this must be taken into account for breeders. Self-compatibility is usual in autogamous species. Okay, the reproductive system of autogamous species usually involves plants which has the two reproductive sexes, male and female flowers, maybe in different points of the plant, or even in the same flower. Okay, hermaphrodite flowers. That favors self-compatibility. Okay. On the contrary, in allogamous, you will find in the reproductive systems and processes several difficulties. Maybe the species is monoic, like mice, but the flowers appears in these different distant points of the plant. The male flower inflorescence is on the top. The female inflorescence appear in the medium of the in the midpoint of the plant. So physical barriers, or maybe the species is dioic. I mean, it has male and female plants. Or even in the case that the flowers are too close in the plant, monoic, or even hermaphrodite, these species have other mechanisms of self-incompatibility, okay? Genes of self-incompatibility, which provoke barriers to the germination of the pollen grains in the female flowers, or maybe post-zygotic barriers. Like for instance, okay, there was not a mechanism preventing the fertilization, but okay, after there are other barriers, post-zygotic barriers affecting the viability of the offspring. at the seed stage or even later when the plants are seedlings, like deleterious problems or weak development, etc. So in allogamous, there are different barriers which prevent self-compatibility, which on the contrary is quite usual in autogamous species. Okay, and I'm saying all of this, and you will see in the examples of Pedro and Barbara, because in summary, flower biology is essential to decide the breeding methods. Okay, in fact, in some cases, hybrids are not economically viable, as has happened for the years, for instance, in wheat, which is highly autogamous, and it makes not economic sense to obtain hybrid seeds, because for each seed you need to make artificial hybridization, so it makes no sense economically. Okay, this is an extreme example. But I repeat, flower biology is essential to decide the breeding method, and this must be keep in mind for future breeders. Well, regarding the main methods, I will be very brief because Pedro and Barbara will provide more details in their practical examples. We can separate in two big blocks. The first block is in such cases where the starting population is heterogeneous enough or shows enough genetic diversity to perform a breeding process, a breeding program, which will satisfy the breeding objectives you have. Okay, I have enough diversity in this population, the starting population. Well, no need of hybridization or introducing new varieties in my program, etc. Okay, in such cases, we have two main strategies: mass selection and individual selection. The mass selection is, they are both quite similar, but the details which differ are the following. In the case of mass selection, in the first year, imagine you have a plot, a field with 100, 200, 500 plants of your heterogeneous population. Okay. In such case, you go this first year and select the best individuals. Okay. I will select 5 or 10% of the best individuals. This one, this one, this one, this one, this one. I label and I wait them to reproduce and get the seeds. Okay. I can leave all the plants in the field. Okay. Or maybe if I want to prevent strictly cross population, maybe I can remove the other plants which have been not selected. Okay. And just leaving the ones selected. It depends on your how strict you are in your breeding process. Well, once I get the seeds from these selected individuals, I mix them in a bulk. Imagine you have selected the 5% of your 500 plants, which means 25 plants, 25 offsprings, 25 lots of seeds, which are then mixed in a bulk. And then next year, I will take seeds from this bulk and will evaluate again in the second year. And in the second year, I repeat the same process. Okay, I have planted 500 plants, and I will identify those plants which are the best ones for my breeding objectives in terms of vigor, in terms of production, yield, morphotype of the fruits or the grains. Who knows? It depends on your breeding objectives. I select them. I take the seeds and I mix it again in a bulk, and so on, repeating year after year. Okay. In this way, I am not tracking. I'm performing selection. I'm being a bit strict because I'm not leaving 90 or 90%, 95% of the individuals to reproduce, but the 5 or the 10% I like the most. Okay, but I'm always keeping some degree of variation because I mix them. Okay, the seeds from those selected individuals. I'm not tracking individually the offspring of each individual. So, and this is mass selection because I mix in a massal way the seeds from the selected individuals. Well, this is mass selection, briefly. Individual selection, by contrast, under individual selection, the first year, imagine again my 500 individuals of my heterogeneous segregating population, I identify the 20, 30 best individuals according to my breeding objectives or traits. Okay, this one, this one, this one, this one. But for the next year, what I'm going to do is to take the seeds individually from each selected individual. And by contrast to mass selection, in the case of this individual selection, I will follow each offspring separately. I don't mix in a bulk. I take the seeds of the individual number 75, which was one of the selected. I take the seeds, and next year I will evaluate individually this offspring, together the others, but tracking them. The same with the others individual selected. You can see here the separated rows corresponding to the offspring of each individual selected individually the previous year one. Okay, and I compare. If the population is heterogeneous enough, maybe I can perform selection in the year two within row because it is still heterogeneous. But usually selections are performed directly by rows, by offspring. So, okay, in this second year, the offspring for the former individual 75 and the former individual 22 and the former individual 178, I don't like. I realize that I don't like. Maybe the environment pretend me in the first or like me in the first year, and they were not so good, and the offspring is not good. But on the contrary, the offspring or the individuals 71 and the individual around 125 and 193, their offspring seen and evaluated this second year are good. Okay, I continue with them, and so on year after year. Okay, so this is individual selection. I'm tracking each individual following their offspring. And now the big block on the other side. I decided that at the beginning my population was not diverse enough to deserve a breeding method itself, or I had no variation with my population or my variety. I need to introduce new variation. How? By hybridization, by crossing with other sources of variation. Okay? These methods start usually by hybridization, by crossing two parents. There are others, I will say, as I will say at the end, like composite cross population, we have seen in Unit 1. Okay, but the most common procedure is crossing my variety, okay, that of my population, which lacks of diversity. Maybe it's good for many traits, but is bad for resistance to a new pathogen, or it lacks for a sweet taste. And I discovered a new source of variation, parent 2, which provides good taste and high levels of sugars and acids. Okay, I cross both them, both. I develop the F1 and I start working from the F1, like Mendel. Okay, and in such case, I have 4 different main methods: the mass selection, the pedigree method and the backcross method. And finally, on the right, the composite cross populations that mix not only two parent lines, but more parents, okay? Well, regarding mass selection, is more or less the same that explained in the previous case. I start in the F1. Maybe if I find some segregation because the P1 and P2 were not pure lines because they were, but they were heterogeneous. Maybe in the F1 I start to see segregating individuals, maybe, or if they are pure lines, I must wait till F2. But the case that usually under mass selection, I perform year after year the F1, then I take the seeds of the F1 to get the F2, which will be evaluated in year 3. Then I take seeds in bulk of the F2 to get F3 seeds, which will be evaluated in year 4, and so on, and increasing the number of individuals from F2 to F5, okay? And then when I achieve a certain level of selfing, then we start the real selection breeding process. Till now, usually we're under this mass selection procedure, I just do growing the plants, recovering the seeds, planting the seeds, growing the plants, recovering the seeds, etc., and increasing the number of plants to arise to get as much diversity as possible, increasing the numbers at about F5 and working about 1,000 of individuals at F5, then I perform selection. It is supposed that at this level the population has increased their homozygosity in many of the genes and the traits, and then it's time to perform a procedure of mass selection. I start to select the best individuals and mixing in a bulk the individuals, and again repeating and repeating the procedure. This is mass selection applied to starting by hybridization. The pedigree method is similar to the individual method explained before, but with the difference that I have started by crossing two parents. The F1 should be quite homozygous, while F2, F3 will show most of the segregating diversity which was encompassed by the two parents. Now it's time to start selection through the pedigree method. During these first phases or generations of selection in the pedigree method, breeders usually select individually the best plants and then track individually the offsprings. And even within the offsprings, in these first stages, or F3, F4, F5, it is allowed to select even within offspring, because the offspring are still so segregating that new recombinations of interest can appear within the same offspring. Okay, even those have been selected individually previously. Okay, and by about F5, F7, then the offsprings are so fixed that then you can go directly to evaluate among strains, among races, among lines, and then select the best lines. And then at this stage is not allowed. It makes no sense to select within a strain, because now they are quite fixed genetically. And this is the pedigree method in summary. Then in the third point, we have the backcross method. The backcross method also involves hybridization, but the objective is, as I explained it previously in Unit 1, okay, the hybrid is not the objective. The objective is to introgress a specific part of one parent, call it the donor parent, in the variety I want to improve, because I want a new version, let's say, a new version of my landrace or my elite variety, keeping all the traits with the only exception of one trait or couple of traits which is given by the donor parent, because I'm not interested in the donor parent, but one or two traits. I'm interested in the recurrent parent, in the variety I want to improve. And only interested in a very small part, genetically part of the donor parent. Then first I hybridize between the donor parent in orange and the recurrent parent, the one the genetic background I'm interested on recover. Then I have the hybrid, the F1, which should have half of both genomes, including the trait or genes that are of interest from the donor parent and the background, and half of the background I want to recover, which is the recurrent parent. With this F1, the next step is to perform a new cross with the recurrent parent. I mean, the genetic background I want to recover. Then I get the BC1, back cross 1, which has 75% of the recurrent parent and just 25% of the donor parent. In this stage, I select those individuals of the BC1 that by appearance are recovering the morphotype of the recurrent parent, the variety I want to recover, but also showing the trait of interest from the donor parent. This basic one individuals, and then use it for a new backcrossing towards the recurrent parent. In the next BC, which will be BC2, I should recover 87, 88% of the recurrent parent. Okay, and I have just 12% of the donor parent. In such a population, I will repeat again the evaluation of those individuals who keep the trait of interest from the donor parent, but with the appearance or morphotype closer to the recurrent parent, to perform new backcrossings, and so on, repeating year by year, generation by generation, till the end, recovering BCC, BC6, BC7, the most of the genetic background of the recurrent parent, but always taking the only part which interests me from the donor parent. This is the backcross method. And finally, composite cross population. As we have explained it in Unit 1, composite cross population are made of hybrids as starting points, but several hybrids. Usually, composite cross population comes by mixing different hybrids. For instance, I have 4 or 8 parent lines, which are complementary for several traits of interest. Then I cross them in pairs to get the F1s, and maybe the offspring of these F1s is directly used to start my breeding program, which can be able to mass selection, pedigree method, etc. Or maybe the 4 F1 hybrids can be combined again to get double hybrids. And then these double hybrids will be used as a starting point, segregating diversity population to start my breeding program. Okay? This is the only difference. Composite cross population comes not only from just one hybrid combination, but from several to start the breeding program. And this is all from my side. As I said, the reproductive system, I repeat, determines the most accurate breeding method. I repeat, in autogamous, no problem on using selfing, generation by generation, but in allogamous, selfings are limited. Now, Pedro Mendes from EPC and Instituto Politécnico de Coimbra, Portugal, and Barbara Pipan, guys from Slovenia, will give you some practical examples of first mice, Pedro, in allogamous, and then later Barbara in common bean autogamous crop, the practical examples of breeding methods using these two crops. So now it's time to give the floor to Pedro. So Pedro, is your time. Please share your presentation. And I think that you will share your screen for your presentation and for be more efficient also Barbara's one or
Okay.
common bean. So Pedro, the floor is yours.
Okay, thank you, Adrian. Thank you for the nice explanation and very, very leverage for what I'm going to say next. As you can see, this is a picture of selection from what we did yesterday in the mass selection that Professor Adrian already did explain. And I would like to follow some of the information that we usually do in our program. Well, I think that this was already stressed by Professor Adrian regarding the allogamic plants that are the same as allogamous or cross-pollinating plants. They exchange of genetic material between different individuals, which enhances the genetic diversity within the populations. So generally, it's more interesting in terms of adaptability and resilience, if we talk about the populations. Well, as it was mentioned before, it's really important that we know what are the plant that we are dealing with. And knowing that, we need to know where is the female and the male part, if they are separated or not. And in this case of maize, we are very lucky to be able to, at least for doing the crosses and all the information, they are big. It's a big plant. It's and they have the separated male and female. So it's really for something for some works that are really interesting. I don't know if you know that the tassel produces around 25 million pollen grains. A year can have 1,000 ovals, the potential kernels, and if you see, this is um, the the pair of male spikelets with 3 anthers dangling from the upper floret in the pedicel spikelet, and then we have the silks that are important for catch or adhering pollen that will be the fecundation and generating the pollen. Well, what are the flowering implications? And I'm showing you these images for you to know about how this works in terms of flowering. This is the tassel, and the tassel generally starts with the main branch that starts to flowering, and then this is going from this part to the other branches, and then it finishes, and it could take from 3, 5 days, sometimes more. It depends on the structure. And in the other way around, you have the silks. This is a turgid silks. They are not getting the pollen yet. And when they are, the pollination occurs, you have this turgidity finish, and the silk get down, and you know that the fecundation already were finalized. It's interesting also because the first silks that appear, they come from the bottom of the year, and the last ones are from the top. Well, you can have several things happening, and in nature you have uh, a lot of differences, but this is generally the normal type. So the flowering allows us to know and classify the type of cycle. If we have early maturing or late maturing cycles, probably you have heard about FAO, FAO 200, FAO 600. It means that they are early maturing types, or late or very late maturing types. Um, the flowering also, as I mentioned it in the last session, in the that can indicate how homogeneity is the population. For instance, if you go for a hybrid that is almost a clone, you will see that they flowering almost at the same time. There's a synchronicity very huge. But if you go to the population, and not a very population that has a lot of diversity, you can see that there's a lot of differences between the flowering time on that population. This is also very important, as Professor Adrian already mentioned, regarding the crosses that we need to do. We can do crosses from two. Imagine that we have two inbreds, and we wanted to cross them, but for instance, one is very early and the other is very late. So to do that, we need to sow in different times in order to make the synchronization with the male and with the female. So it's possible that we need to do separate dates for doing these crosses. And to do that, all these studies, we need a possible, if we are crossing different inbred lines or even populations, we can have hybrid populations. You need to know what is the flowering time. Well, in these slides, what I wanted to show you is what are our main activities, um, because we have some activities that are on the field, and the other activity, sorry, uh, all of them are on the field, but one are activities on farm, and other activities that are on station. So when we are talking about on-farm trials, we can talk about the selection trials. And this in these selection trials, what generally are we do, or with the farmers, or the farmers, if they are instructed, they can do it by themselves. That's what we generally want that happens, and that you can have farmers that they have their own germplasm and they want to improve their own germplasm. We don't want to impose some of the varieties that we already have if they want to continue. What we say is that uh, if you want, we can help you thinking betterly how you can improve your selection on your field. So we generally use the max selection, as Professor Adrian already mentioned, and we also can give some germplasm to the farmers if they are interested in some type of germplasm that we do. We They can ask us, and uh, they start the trial and the mass selection process, as already was mentioned for mass selection. What I would like to tell you is regarding the mass selection, we can do it stratified mass selection. What is the difference of stratified mass selection for mass selection? Well, if you remember about the slides that Professor Adrian showed you in mass selection, you saw that there was a mass selection that you select one plants that would be the progenitors uh, for the other cycle, the other year, for instance. But in our case, we need that we have different field different sites on the field. So it means that in the field we can have a field that have, for instance, higher nutrients, other that is medium nutrients, or and the other it's very low nutrients. Or you can see as another stress option. And when the farmer do the selection, if they don't know about this, they will do the selection only in the good part of the field. They the, they don't search for all the variability that can exist and how the plants tackle with these problems. So the propose is that you do the mass selection, but in different parts of the field. So it means that you will obtain the best years or the best plants in the best soil, the best plants in the medium soil, and the best plants in the worst soil. And then you do a parallel selection, because some of the years that are in the bad soils are can have a phenotype that are equal to the worst years that are in the best soil. Okay, so this means that we need to do parallel selection to have a stratified mass selection. Well, regarding this was regarding about the selection. And I have here trial varieties. Why we do the trial varieties? One it's for to show to the farmers that there are different varieties that they can make their options, and they can use them in their fields. Other trials are the trials of the cycles of selection. Why these cycles of selection? Because during the mass selection procedure, we from one year to the other years, we cannot say um, very strongly that, oh, there was a big improvement of yield, but we need to measure that. And to measure that, we need to store the seeds from different cycles. So this means that if the cycle 0, you need to collect this cycle 0, and then collect the cycle 1, collect the cycle 2, put it in the storage, and for instance, 5 years after, you wanted to know really what did happen. And for that, you go to the refrigerator, you pick the seeds. Generally, you do the regeneration of the seeds, and then you do the trials, because you want to compare what did happen in this process. You can do this in the by genetics, by molecular genetics, or simply in agronomic way. You wanted to know about that. This is the ABC phenotypic recurrent selection that we have. And as you can see, we can do the selection. Generally, it's very seldom among farmers to do this, but you can do what we call negative selection. In this case, you go to the field, you take the tassels, and the tassels before pollination occurs. Okay, this is very important. Otherwise, it's useless to do that. You go to the field, take the tassel before pollination, and take the tassel from the plants that you don't like, or they don't respect the archetype that you have in your mind, or other. This means that doing that, you improve the population, but you don't lose the grain, because these plants will receive the pollen from the other plants. So at the end, because they don't have tassel, you know, okay, this is a good year for grain, not for seed. And the others, you can proceed with the selection. And this selection is defined. Generally, we do it with a farmer. Say, what are the traits that are really important for you? And generally, we also talk with them. And for instance, they suffer about the root lodging, stalk lodging. It's really important, that one. So we need to stress on that. And then sometimes if it's prolific or not. And also quite important is the where the insertion of the ear occurs. Where is the site in the plant? It doesn't. We don't want that the insertion of the ear will be too high, because it makes a big effort to the plant to don't break, and it's much harder for the farmer to collect these ears. Um, Other aspect could be the angle of the leaves. Um, well, many other traits, and of course, uh, we look at the plant, and if, as you see here, the man kicking the plant, because this is connected with root, sorry, with diseases or quality of the root, because if the plant is not good, it will breaks. And so we don't want the seed from the next generation. This is what we do in the harvest in the field. Then we bring the ears to the storing facilities, and we look at the ears, as I mentioned it in the first slide. Sorry, again. This was the slide where you see all the years were disposed, and then we were doing the selection about what will be the best years for the next generation. Sorry about this. And then we select, like for instance, the size, the row number, the health, if it's determined or indeterminate, whatever. After you select this information about the year trait, about um, also about if you select for stand, it's very important in all the selection. It's important that you make what are really your goals, and you should fix your goals, uh, because you cannot change from one year to the other. For instance, if you want to increase the stand, it means that you will have much more plants per square meter, and the plant, and if you have diversity in your population, this will happen. But you cannot do it in one year. I increase the stand, the other year I decrease the stand. It doesn't. it doesn't work like that, because you are making also in terms of genes, you are doing the selection for a certain trait. And if you move and if you change very quickly, it doesn't, it will not be nice. Other aspect regarding the trials that I've already mentioned, and we talk about with the farmers, it's how you are going to do this. It's really important that you have a border. Border are really important for trials, because it could influence the pollination. It could interfere with pollen from outside, because you want to have isolation. You want to have your field isolated. So how can you do this isolation? You can do in time. If your neighbor have the same cycle as you and he much earlier, you know that you need to wait for avoiding cross pollination. Other, it's the isolation, the kind of physical isolation. You know that in that place there's no other growers, maize growers, so you are relieved on that. Also, a kind of very important, if we are talking about the selection in the organic, or even for your local adaptation. And as you can see here in the drawing, you have the seed in the center. It means that these seeds are done, for instance, in conventional. You buy the certain hybrid, and they say you should do this, and this in order that you achieve or you maximize your yield. In this way, you and in this case, the breeder or the seed are the center. They define what you are going to do. In this case, and that's why uh, this is important in organic, because you need to adapt for each place in certain cases. Obviously, there's some varieties that could that could be good across the locations, but others that are more local adapted. And in this case, the farmer is the center. So he takes the decisions, he makes the decisions, and he adapts the variety that is better for their eco region. In case of conventional, it's you have the seed. So what you need is to tune to achieve the best as you can. But if you have um, different land, different regions, or you can tune them with more fertilizer, with more amendments. Well, when we are thinking about trials, and we want to really to know what is the value of our varieties, you can have several trials, because this is the way that you phenotypically can go and can understand better the genotype also, and understand if a certain variety is really behave very good for all the locations that you want, or uh, it it is specific for a certain location. It behave very well, for instance, in this region, but it is not very good in this region, or could be in average good for all these regions. And what about the on station? What we are doing in the on station? Well, generally, uh, for instance, for new combinations, as already Professor Adrian mentioned about crosses, in some cases we wanted to do some diallels. This means that we need to cross, if we have, for instance, 10 populations, and we want to know if we can do some hybrid population, because it's possible. If you have two fields, you can have a male, and in the other field you have the female, and in the field with the male you select for the male, and in the field for the sorry the field for the male you can have the male and the female. What is important is that you can't avoid that the tassels of the female will appear. And in this case, you can select for the male, and at the same time, you produce the hybrid from the two populations. And in the other place, you will have the female where you are selecting for the best females. But when we wanted to do the diallels to know what are the best combinations, this means that we need to cross one, for instance, if we have five, one with two, one with three, one with four, one with five, and etc. Then the two, well, we have all the combinations possible in order that in the next year, after we obtain the hybrids, we will see what will be the behavior of these hybrid populations. We can have also some dynamic populations, synthetic populations, and then we do also the selection in the, for instance, in the populations, in the synthetics, etc. We can use the recurrent selection by S1 or S2. This could be also in the farmer field, but for this you need to instruct, and the farmer needs to be really excited about doing this. Then the varieties, you are going to test also, as we did in the farmer, the germplasm. It could be also a way to show to the farmers the value of certain varieties. Um, you can test also what is the cycles of selection, what are the degree of this, what are your gain of selection, and do all this. And also doing the maintenance in the nursery, you need to maintain, you have all the cold storage facilities, but it's important that for some years you refresh, for instance, the inbreds and refresh the populations. And it's important, for instance, inbreds, you should make the selfing of inbreds to maintain the inbreds. You cannot cross them. But for instance, for populations, you need to cross them to maintain the variability. You shouldn't do the selfing with the populations if you want to maintain the populations. Obviously, that you can self you if you want to obtain other stuff. Well, this is basically what we do when we do crosses. First, you need to go to the field and ensure that the ear are receptive. And this is the very um, uh, late stage, because you need to put this bag, that could be a plastic bag or a paper bag, you needed to put it before any of the silks come out. If theirs come out, it's finished. You cannot use that plant. And then if you want to synchronize, because remember that are one that come before and other that come later. If you want to synchronize, generally we cut the ear, the tip of the ear, because this is also interesting, because the silks will necrose and avoid that pollen, for instance, to germinate. This is generally what happens in the next day. So this is what are you doing in the female. And what about the male? Well, in the male, you have the pollen here. You see that already started the pollination, and then you uh, close the bag, and In the next day, you beat the bag and you do the pollination. And in this case, you know that this that you did a cross or a selfing, because with this simple, very simple, you can build a very complex and strategic ways of breeding. This is the field, how it behaves during the pollination. Um, it's important sometimes of the physiology and the other studies are very that could help us really uh, very much. For instance, before we knew that we didn't know about if we when we were um, preventing the pollen from outside to be inside. So when we do this, the question is, do this tassel is full of pollen from the outside? And if so, there's a problem when you do the pollination. But what we knew after is that the pollen has a 24 hours. The duration is less than 24 hours. So this means that if you have this from from one day to the other, the pollen from the previous day is almost gone. So you will not have problems of other pollen that you don't want that be there. Well, at the end, you come and you select, you take your ears and you monitor your ears, for instance, indicating what is the time that the pollination was made, what is, for instance, if there was prolific or not, what is the number of nodes, and etc. This is the story of a synthetic pollination, but if you really wanted to know, I will let it for the model, the unit 5. I hope that you come to it. Well, this is the other the way that you do selection, but in this case with S2 lines. Why you do this? Remember that for allogamics and especially for maize, it don't like at all to be selfed. So when we self this pollination, these populations, it means that they are inbreeding and certain genes start to be recessive and start to emerge some deficiencies, even white plants, etc. So this basically the system that we do is doing around 1,000 S1s, the selfing. Then in the next year we do 500 to 600 S2. So we self again from these self ones that were chosen. Then we choose the 200 selfing tools and we do a trial, evaluation trial. And in this third year that we do the evaluation trial, we select what will be the best 35 elite S2. And with this 35 elite S2, we make the recombination of them. And we pass from the cycle 0 to cycle 1. And we do it all over again. And in this way, we can ensure that we have some inbred populations, a breeder improved populations. Well, for you to know that our varieties are irreverent. You see here the photo, the mostly hybrid maize, and in this case you have the different vegetation habits, as you see here, in comparison with the hybrid. I mentioned already in the previous session about the way that we could in some way using some methods for selection. For instance, if you have a lot of material, you can do a very fast selection using the eye to uniformity, angle, tassel, ear, and root lodging. So as stalk lodging, this here is the position of the plant. So you consider the in average the plot how is it behaves. And yes, we can easily also connect with other breeding methods. We have done some trials, even trials with regarding the yield, because even that we have a more accurate when we do trials, obviously, we wanted to uh, have an idea about how farmer can consider their yield and if they could use some of these tools with us and then with in their own farm. So thank you very much for attending, and please, Barbara, the floor is yours, and please tell me when you want to that I move the slide.
Okay. Thank you very much, Pedro, for a very nice presentation.
Thank you.
I'm Barbara Pipan from the Agricultural Institute of Slovenia, from the Crop Science Department. And now we are actually moving from maize to common bean. And with this, I will start because this picture is very nice and very informative. And this is a logo from World Pulses Day, which is every year in 10th of February. And it's actually a very descriptive picture about different pulses. Within those, also common bean is one of the most important. Please, next slide, Pedro.
Mhm.
Maybe just a little bit of introduction about the common bean. It's actually one of the most important edible legumes. It also belongs to the group of pulses, which is for the human consumption. And it's a valuable source of proteins, carbohydrates, fibers, and rich source of other components with nutritional and health benefits. For the Slovenian production area, common bean is known as vegetable of increasing agronomic interest. So Slovenia is the middle of Europe, and as well our people and ourselves are very keen to grow beans, to cook beans, to use beans, and to eat it in any possible combination. So that's why it's very important in this Central European part for different purposes. So maybe if I will focus only about Agricultural Institutes of Slovenia, in the past, 4 different varieties of common bean were developed. So Adrian before nicely explained different types of or different methods of the breeding, and these 4 varieties were actually developed using individual selections. Selection from autochthonous native populations from Slovenia. But in the last decade, we started with hand cross pollination, so with targeted hybridization and pollination of different types of bean for different purposes. So with this, recently, we are introducing the organic breeding as well in parallel with conventional breeding. So this presents also for ourselves a huge challenge, and we are actually very keen to develop this and also to provide the new varieties which are not only suitable for the organic production, but also that they're organic varieties itself. So please, next slide. Okay, so it was nicely explained everything about the breeding, the plant breeding, and I will really go from the scratch and focus about what actually plant breeding is in very how to say it in very simple way. So one of the definitions of the plant breeding is that this is the science and art of improving plants to produce desired traits. This involves selecting plants with favorable characteristics and breeding them to enhance qualities such as yield, disease resistance, drought tolerance, and nutritional value. So the objective, the goal is to develop new plant varieties that are more productive, resilient, and beneficial for agriculture and horticulture. So from this point of view and these objectives come our ideotype. So something refers to an idealized model of a plant that possesses a combination of traits aimed at achieving maximum yield and quantity within a specific environment. So ideotype is actually something we want to reach. We want to get from already present germplasm, available germplasm, and in which direction we will improve it through the breeding program to reach the ideotype. And ideotype is strongly associated with our breeding objective. So, and actually an ideotype serves as a blueprint of these characteristics, guiding breeders and selecting combining traits. So by targeting these traits, breeder can create plants that are more efficient and utilizing resources and better suited to their growing conditions. So with this, it's very important that in consideration with the ideotype and was actually the growers, the farmers, the consumers wants is strongly associated with the breeding objective. As Pedro already mentioned in his presentation before, it's very important that we have them at the beginning and that are clearly stated and that follow them along with the whole breeding process. So next slide, please, Pedro. And what are the breeding objectives in our common bean breeding program? So as you know, I'm sure that all of you know common bean very well, and we all know that common bean is actually of a different growth habit. So we have dwarf types, we have climbing ones, and we can use beans as fresh pods, we can use beans as dry beans, and with this different appearances of the beans also comes different breeding objectives. And our main actually I've listed some main breeding objectives which are the most important in our breeding program. So first, already mentioned before, is tolerance to biotic and abiotic stresses. With biotic stresses we have in mind the most important diseases, and also one pest, bruchid, which is very of economic importance when growing common beans. And with the abiotic stresses, we have in mind temperature stress and water stress. And earliness is something which is very important in our breeding program as well. So that the varieties which are developed are early enough that they can avoid during the flowering, they can avoid these high hot temperature during the summer, which are actually then the consequence that the flowers drop out from the plants. And if the flowers drop out of the plants, we will know that there is no yield at the end. So this is one of our main objective as well. And for the others, mostly for the snap beans and beans of climbing beans, which is used for the fresh pots. This Central European area is very keen to use. So I'm not sure that you are familiar with the word maslinets, but it's actually a type of the beans which has a very long, flat, yellow pods without strings, and is very common in this hot summer during the hot summer for the salads, and it's used for the fresh pods. So these types of beans is very desired and beneficial as well for our area. And of course, for the using the machinery when harvesting the yield, it's very important the position of the pots on the plant, especially for the dwarf beans, so that we can use the harvesters to pick up the yield at the end. And of course, as all of us know, the highly yielding varieties are also very desired within different breeding programs of different plants. And recently, we are really focusing on the favorable nutritional composition of pods and seeds at the end. So with the higher levels of proteins, iron, zinc, and decreasing values of the anti-nutritive compounds such as phytic acid. Next slide, please. Okay, so this slide is actually the summary of the whole breeding workflow of the common bean. So maybe before I didn't mention that all the pictures from this presentation are from our breeding activities. So we're taken through our breeding activities, and that common bean is also a member of the Living Lab, which was established within the LiveSeeding project at Agricultural Institute of Slovenia last year, as well as potatoes and buckwheat. So we have also at our institute national public breeding programs, not only for common bean, which is smaller one, but as well for the buckwheat and for the potatoes. So now we will go through the breeding workflow, which starting with the parent selection. So parent selection, we will go more in details each specific step in the next slides, but just to have everything on the same level. So first is the most important is the parent selection, and then when we have the parent selection, so we have mother, we have father, we do the hand pollination, so targeted cross pollinations, targeted crossings using tweezers and all the equipment needed for that. And then after that, we are doing the selfing and the selection at the same times. And then doing this phenotypic selection during the whole cycles of selfing and genotypic selection, so marker assisted selection in F2 generation. And after 6 cycles of selections, so phenotypic, genetic, and then the selfings under protected conditions, of course, everything, all those steps are under protected conditions, that we are not having any unwanted or uncontrolled gene flow from the outside, so that we have everything under control. And then we come with the final selection to select the lead breeding lines, do some additional in vitro tests and other open evaluations, other evaluations on the field, along with the parental lines and standard varieties. And then after that, we decide about the best performing elite line and put it into the registration. So, and for the common bean, this whole process from the first crossing, not from the parent selection, but from the first crosses to the final registration for common bean is approximately 10 years. So now here we will go very briefly and through all the steps of the breeding workflow for common bean. So Pedro, please, next slide. Okay. In the module 1, Vladimir was talking about pre-breeding. So these activities are very important when we are about to select the appropriate and the most suitable parents for the breeding. Of course, the selection of the parents also is in a strong association with the breeding objectives. So because if the parents does not have the traits of interest we want to improve during the breeding program, also our candidate breeding lines cannot have it. So that's why this step is crucial and very important. And very important is that we have knowledge about all this germplasm, all this variability, Adrian talking about all the variability which is available and could be exploited and improved through the breeding program as well. So that we know if this material can be identified, that we know what we actually have stored mostly in the worldwide gene banks, which are the treasures of the diversity, especially for the common bean, and then we combine the germplasm from the mother, from the father, and to actually get the best performing clients in the end. So it's very important that we have all those materials characterized so that we know what we have, and that at the end that we have all these materials available so that we can actually create and have the possibility to grow them and then to do the crossings then in the further steps. So that's why I put here the importance of the identification of all those germplasm which is available. So here we are doing a lot and collaborating a lot within different European projects which are dealing with the characterization and evaluation of the materials which are stored in gene banks and elsewhere around the world, that we can use it in the breeding programs as well. So before I have mentioned the hand cross-pollination. This is very delicate activity, which is of course done in the screen house under protected conditions during the flowering period of the common bean, which is the end of June and the whole July. And here is very important that we have the synchronized flowering, because all those parental line are different and are of a high variability, and they don't flower at the same time. And very important that we have available different flowers from different fathers to do the crosses on the same maternal plant. So because we do the crossings with different fathers on the same maternal plant, it's very important that we have a system how to label different parents. So all those labels have different lanes of colors for the fathers, and with this we can see which father is actually done with the for the crossing. And this crossing itself, it's very as I said, very delicate. It needs to be a lot of concentration to put it, that you actually open the flower on the maternal plants. When the flower is open, the flower is open, it's already self-pollinated. So we have to open it before it opened by itself, that we can transfer the pollen from the father flower to the mother flower. So it's a kind of procedure. And then when we do these hand pollinations, we are actually hoping that that we were successful and that we will develop viable seeds at the end of the growing season for F1 generation. Next slide, please. Okay, and then, as I mentioned before, there comes self-pollination, phenotypic selection, and marker-assisted selection. So everything is done under protected conditions, and self-pollination and phenotypic selection along with the traits of interest, so that we see all different plants from the same combination about their phenotypic traits. And in the F2 generation, we perform the marker-assisted selection using the panel of 24 different functional markers. So we would say DNA markers, trait associated markers, to actually check the genetic background of some of the important traits. So if they have, I don't know, resistant genes to fungal diseases, to bean common necrosis, mosaic virus, which is most important in common bean, to Xanthomonas, and all the others important traits where the markers are available and possible to screen. So about this topic, I will talk about in the module 4 in 4th of March, more in details. So in combination of the results of the marker-assisted selection and phenotypic selection during the different cycles of selfing, we then decrease the number of the plants which are suitable and which are actually the most suitable for as a candidates and the most promising for the registration at the end. And then we have a really, I don't know, some the most promising breeding lines, and then they go to the open field performance, and we did do the additional in vitro tests. So please, next slide, please. So open field performance is always done under randomized blocks in 3 repetitions together with the standard varieties and along with the parental lines, and additional in vitro tests are required to describe the variety and the susceptibility to the most important diseases of common bean. This is bean common necrosis mosaic valus, fungi Coletotrichum lindeomuthianum, and the bacteria from the genus Pseudomonas and Xanthomonas, because all of those data are at the end needed for the fulfillment of the requirements which are needed when we submit the candidate variety for registration to the Community Plant Variety Office, which is independent office for the variety testing in France directly, or from or through our national ministry, agricultural ministry. So here it's very important because those tests are tests if the variety is distinctive, uniform, and stable. These are called DUS test. This is called DUS test, and this DUS test is actually done for each plant candidate variety. For common bean, this is 2 years. And then after that, we then get the results from those observations, those tests, and see if the variety is then registered and protected from CPVO. Is protected under European Union, and as well then listed to the national list of variety and European catalog of variety. And this is a special procedure for common bean and for the other species, where all the data should be available and concentrated at the same submission paper. Next slide, please. For the moment, KISS has from this hand cross pollination, so coming from hand cross pollinations and targeted crossings. At the moment, we have 3 new varieties from 2021 and 2023. We have KISS Amand, KISS Marcellian, and KISS Pauline. All of those are of a dwarf types. KISS Pauline is for the combining use for fresh pots and dry beans. KISS Marcellian is perfect for the dry beans, and KISS Amand is perfect for the fresh pots. So those 3 varieties are not organic, but are can be used under organic as well, because they are well adapted to those low input conditions. And we also have 2 organic heterogeneous materials, KISS Deodat and KISS Bogo, which are both heterogeneous. So KISS Bogo is heterogeneous on the pot shape, and KISS Deodat is heterogeneous and of the seed coat pattern. And 4 candidate varieties are still under the DUS testings as well, so at the different stages. So maybe here would be also interesting to expose that the denomination of the variety at the end, because here you saw that when we submit the candidate variety to the variety office, we supply breeder's reference. So this is a kind of a mark and label the name of the proposed variety, and we also have to propose the denomination. So we have KISS Amand, KISS Marcellian, KISS Pauline. So this KISS prefix is because it's Agricultural Institute of Slovenia. So in Slovenian language, Kmetijski Inštitut Slovenija. So that's why it's KISS. And then for the common bean, we have the unique key how to select the name of the variety, because it's strongly associated with the flowering period. So when the full flowering occurs for the candidate variety, and if the sowing date is 16th of May, as it as a kind of traditional orientation in Slovenia that it's own, then we just look at the calendar and see which male name is actually on this day, and we decide how to will select the name of our new variety. So this is just something for the additional information. Next slide, please. And yeah, as well, since I didn't mention, I'm also not just a common bean breeder and leading our national common bean breeding program, but I'm also a researcher from the field of genetics, molecular biology, and GIS-based applications. And since it's very important that we know that a lot of data are generated through the breeding programs and the whole breeding process for the legumes, for the common beans, and it's really very nice that we can that we learn how to combine the different types of data and to present different types of data also on scientific way to through the different projects which actually give us an insight about the basic knowledge and how to then this basic knowledge can be published scientifically and at the same time transferred to the breeding program to increase the applicative value, because this is very important that the same people are working on two sides and to transfer everything which is useful and can be exploited and further developed in the applicative story like is breeding. So with this, this is also something which is for the additional breeding, some of our recent publications which are associated with a common bean and legumes. Next slide, please. I wanted to list also some main challenges for the future. So as I already mentioned, it's very challenging to find the elite germplasm, to find it and to make it available. And of course, to address the breeding objective according to the agroecological changes we are facing with in recent years, and to employ new breeding approaches. Since we are inorganic, we are limited to the traditional breeding techniques, and we really want to accelerate the breeding process on the most appropriate way. And that's why the new breeding procedures and techniques which will help us to not increase, accelerate the breeding process are very welcome. And of course, to implement organic breeding in parallel with conventional breeding. And you know, when we have in December workshop together with the colleagues from Serbia, from Zimun Polje, about the breeding within the frame of the Credit Vibes project, its twinning projects, we also talked about challenges in breeding in general, and we actually identified that the most challenging is to find the competent human resources for that. So I'm really very trying, since we are institute, we are not university, to host students here from our university to see our breeding programs, to see our materials, to see our lab, maybe to motivate them and to be involved in our work after their studies. And of course, recently there is a lot of information about open science, and open science, one of the pillars of the open science is also citizen science approach, maybe how to include citizens in the breeding programs as well. And yeah, this is also one of the challenges for KISS as well, maintenance and marketing of the developed varieties. So that when the variety is registered, so after 10 years, that still stays like it was registered, so that is stay stable and uniform as well as it was described. So this is also one of the challenges for ourselves. Next slide, please. Okay, so since this breeding program, as you can see, it's very broad and take a lot of attention. So this is not work just for one, but a group of people who are somehow involved in the breeding process at different points. So here are our colleagues which are involved in the breeding of the common bean, and as well list of the logotypes of the projects, ongoing projects, which are somehow associated with the breeding program at KISS. So I have to mention that public breeding program of common bean is also financially supported by our agricultural ministry, and all the other, of course, scientific or research projects which are ongoing on national and international levels. So with this, I would thank you for your attention, and I guess it is time for the quiz, and I would kindly invite Pedro to continue with the short quiz.
Thank you, Barbara. I will share with you the quiz in order that you can do it. It's already on the chat, and you can also make the reading on the QR code, and let's do it. Good luck. Hello, everybody. Should we continue? Or there's well, I'm thank you for all of you that already submitted. As you can see, there's a big diversity in our sample from Bosnia, Ireland, Belgium, Portugal, Spain, Italy, Poland, Nigeria, France, Romania, Germany. There's a really and sorry if I forgot some. There's really a wide assistance of you, and I will go to the other questions. So the why certified mass selection is preferably yes, and the chosen for farmer selection than recurrent selection by S2 lines. Yes, it's because it's less complex. You can do it. We have an example where breeder have their work on the farmer field with the farmer. I let this for our last session today, the 3.4, but it's less complex. Then should I follow the same methodology for conservation of inbreds and maize populations? As I did explain, for instance, for maize, no, we should not do that because when we are selfing, we maintain the same genetic information, and we want to maintain the pure line, the inbred. But in case you have the in case of you want to breed for the populations, you need to maintain all the diversity that the population has. So you need to make the cross chain one with the others. So maize tassel produces very good, excellent. It's 100% right, good. Can farmers produce population hybrid seed? Yes, it's possible that it's if they have the two fields, as I explained, one for the male and producing of the seeds, and another field for female. Well, what is this is from Barbara. Barbara, do you want to give us some hint?
Yeah, it's actually nicely presented that this is the US test, which is the correct answer. And yes, and all of course the what this actually stands for means distinctiveness, uniformity, and stability of the registered variety. So this is
Very
correct.
good. Thank you.
Perfect.
Do you want to follow this one also?
Yeah.
Okay.
So we were talking about here around approximately 10 years. So of course it could be more, but it couldn't be less. In the traditional way, it's actually not expected to be less than 10 years from the first crosses, because we have this testing for 2 years, and we have to first get the F1 generation, and then to make this generation selfing and do the selection, and this make this population stable and uniform. I don't know, until F6 generation, and then we have some open field experiments and everything, and it's 10 years at least, or 9-10 years. Yeah.
Thank you, Barbara. And
Yeah, super perfect. Yes, it's really strongly associated. So the majority of the students actually answered correctly, and yeah, this is something which is strongly associated with the with each other. So yes, breeding objective and traits of interest are something which is, but not always. So maybe some the answers didn't are completely correct as well.
Okay. Oh, sorry, this is not. Sorry. Okay. This was the last question. I think I should pass the floor to Adrian. Or to Petra.
Okay, no, thank you. Thank you, Pedro. Thank you, Barbara. Such a good and synthesized presentations. I think that your examples were well explained, and show the most applied part of this unit. So it's also good, and also provide a more dynamic environment, learning environment. So thank you very much for your contributions. And from our side, there's no more. I think that you can stop here. I'm not sure if the homework has been exposed. The homework, Pedro?
No, not yet. So the proposed homework is you can use also the link. It was the 10th question. If you have a maize population allogamic that you want to maintain, what strategies can you apply to maintain your population from contamination?